All right, what's up, everybody? How's it going? So, today's class will probably be pretty short. I'm just going to check in with everybody, see how things are going, see if you have any questions about the experiments you're doing, how the experiments are going, all that good stuff. Up until this point in the class, you should have learned all the skills required to perform the experiments in the class. So, uh, yeah, not that there's not more things to teach you. There definitely are, but this class is more of a hands-on class. So, we are here to help you figure out how to get your experiments working. So, everybody should have all the materials they need, right? You should have everything you need. If you don't have everything you need, make sure you post in the Google Classroom or email us, email me, email Esther, and let us know what supplies you don't have. Otherwise, yeah, how's it going? Let us know. So what's going on, Chris? How's your experiments going? You running into any troubles? Sorry, there's a lot of lag on here. So it'll take me a minute to see. What's up, Matt? How's it going? How's your experiments going? Hit me with it. Let me know. I'm ready to help you guys troubleshoot. Hey, Buzzwordy. You're still waiting for stuff? What are you waiting for? Do you live in the US or are you international? If you're international, um, check your tracking number, see what's going on with it. If you're in the US, that's not good if you're still waiting for stuff. Make sure you email me or Esther. So you can email either Esther at the-odin.com or Odin at the-odin.com. Petri plates. Buzzwordy. You ran out of Petri plates? Oh yeah? That's unfortunate. Have the Petri plate shipped? Have you requested the Petri plate ship? Oh, I got most of my stuff but the plates. Did you let anybody know at the Odin that you haven't got your Petri plates? I don't know. Can you DM on YouTube? I don't know if you could DM on YouTube. All right, how's everybody else's experiments going? Anything? Yeah, buzzwordy, just email us, odin at the-odin.com or Esther, Esterther. Oh, Peter knows? Okay. Did you get a get a tracking number yet? Kanamycin, I don't think there's amoxicillin, it's ampicillin. Yeah, I don't think there was any G418 with this kit because you're working just with bacteria. Um, it should be just kanamycin and ampicillin. Plates. here. Here's a link how to make agar plates. Yeah. If you can't see the links I'm posting, change it from top chat to live chat. So at the top part of the chat, in the upper left-hand corner of the chat, there should be a box where you can change it from top chat to live chat. Just change it from top chat to live chat, and you should be able to see the links. All right, what else? Who else has problems? Let me know. What problems can I solve for you? Nothing? Come on. I know some of you got some problems. Got 99 problems. Okay, Joe says he sent 2 sets of plasmids to GeneWiz, none worked. All right, let's work through this. So Joe, did you run a gel and did you see bands in the gel when you ran a gel, or no? Yeah, so if uh, if you don't see any bands in a gel and you send off the plasmids and nothing gets sequenced, um, the next time I would make sure that there's bands in the gel. One big band in each lane. Do you have a picture, Joe? Do you happen to have a picture of that you can send us? What you mean by a one big band in each lane? So sometimes what happens, did you run a ladder also? Could you see the ladder? If you forget to use TAE buffer instead, and instead use water with the agarose, sometimes what happens is you don't get good separation or any separation, and it just looks like a smear or a mess where you won't see anything. So always remember to mix the agarose with TAE buffer and not water. And make sure you run the gel in TAE and not water, right? If you don't see anything, not even the ladder, it usually means you need to add more dye. You didn't add enough dye or something like that. If you see the stuff staying in the DNA, the DNA staying in the wells, right, with the dye, and you see the dye moving through the gel, then it's a good start. So if you don't see bands, the first thing to do is to go back and I so generally what I do is if my experiment doesn't work, if I'm trying to purify 3 plasmids, and thanks Samurai Jack, I'll keep up the good work. I'm trying to purify 3 plasmids and none of them works. I'll start over and I'll only try to purify 1. You uploaded pics to our chat site. So you mean Google Classroom? All right, let me check the Google Classroom. Let's see. I don't see anything on the Google Classroom. Can you post a link or something to the pictures? Maybe put them up on Google Drive, someplace like that where I can get access to them. Yeah, I can't see. Yeah, if you can get a picture, a link to the picture, and send it to me, I could look at it and give you a better idea of what's going on, right? Did you make sure you specified T7, the T7 terminator or reverse primer? Posted on our class stream. All right, on the class stream. Still don't see anything on the class stream. Maybe it's taking time for it to upload. Or did you post them a while ago? Because I don't see anything. I don't see anything recently. Oh wait, let me check. Stream. Yeah. Oh wait. Here, James Dart. Let's see. 4 after, about 1 hour later, running batteries. Final gel. Oh yeah, the gel looks rough. It's really hard to tell what's going on there with that picture. Did you make sure that you created the gel with TAE? That's a big question. Did you create the gel with the TAE? So here's what I do, Joe. First thing I do is choose one of the plasmids. A good one to pick is probably just the GFP. Okay. Now, when you regrow it up and you grow up, say 5 or 10 mLs or however much you grow up, what you should do is when you spin it down, don't just spin down one microcentrifuge tube. Okay. So spin down 1.5 ml bacteria, dump it out, spin it down again, dump it out, add some more, and spin it down again. So try to spin down about 3 or 4 tubes full of the bacteria inside your microcentrifuge tube. That will allow you to purify more DNA. There's a good tip from David. He says, what helped me get better gel results was only invert the tube instead of shaking vigorously on step 6. So only invert, don't shake vigorously. Try that. See if that helps also. But yeah, your quantity of DNA is related to the amount of bacteria, right? So the mini preps can generally only handle uh, about 10 mLs of bacteria total. But what you can do is it really depends on the amount of stuff that's growing. And so just try a couple different amounts. If you need more columns, if you start running out of columns and buffer, let us know, and we can try to send you more for the cost of shipping or something. But yeah, I would try a more amount of bacteria and see if you can purify it, right? Hey, me, myself, I am doing pretty good. Just busy working. Got back from China. Was there for 8 days. It was pretty crazy. Yeah, more bacteria, you know? Generally, this is not always true, but generally in science, if it's not working, just add more. You know? It's not always true, but usually when you're trying to get something done, the most common thing that goes wrong is you didn't add enough of something. So the thing that is inside the column is actually just silica, right? So it's just silica column. That's it. Um, you could probably find, the same columns or the material that's used in the columns anywhere. They're pretty cheaply made. Look for them on eBay or Alibaba or someplace like that from China. Afford your nuance? Yes, we are just troubleshooting and shooting the breeze tonight. Trying to get people to uh, get their experiments going and figure out how to get them going. Oh, you talking about my t-shirt, Anatomy of Next? Oh yeah, one of my friends, Mike Solana, he's the one who makes the Anatomy of Next podcast. So if you want to listen to an interesting podcast about Mars and space and science and startups and a lot of cool stuff. So you can't sterilize the columns and use them again, but what you can do is you can try to wash them and use them again. So what people do is if you take the column and rinse it out really well with water, distilled water, and then spin distilled water through it like 4 or 5 times. What that should do is it should remove most all of the DNA. Now, you know, if you're doing a really important experiment, I wouldn't do this because you can generally get columns online or something for not that expensive. But if you're trying to get these experiments to work, if you're trying to get some sequencing done for your test experiments, then yeah, you can reuse the columns, right? So just rinse them out with water, right? So make sure you get any gunk out of there. So rinse them out good, squirt them with the pipette, and then add some distilled water and spin it down a few times. So let the water go through the column, and then just discard the water, right? Discard the water that goes through the column. And because the water elutes DNA, it should, hopefully, if you spin down enough water, elute all the extra DNA that's sitting around on there. Of course, again, it's not always the case. Is it possible to refine this even further? All right, so you just posted your best results. You see all three samples, but the third doesn't come up great in the pick. All right, let's see. So, yeah, no, I mean, those results look pretty good. So I'm looking at the image you just posted on Google Classroom, and it looks, so what you can do is, you know, you can use a little bit of image manipulation. So if you save the image and open it up with like some image processing software, and then change the contrast, sometimes you could see the bands better. But understand that, like, don't, like, a lot of things can go wrong with loading your gel, right? So what can happen is, like, some of the stuff can get out of the well when you add it in. Like the DNA could be a little bit too diluted. There's a lot of different things that can happen. So if you see something, anything, uh, then you should be able to, generally there should be a plasmid there. Now, if you're going to go on and do the next experiment where you do restriction, digest, and ligation, you probably want a little bit more. You probably want to be able to see the plasmid before you do the restriction, digest, and ligation. But I bet you can send those off even with the weak bands, and you would get some sequencing results back. So, T. Williams, yes, so generally the buffers are really simple. You can look up buffer recipes online. So if you wanted to make your own, right? So you can look up mini prep buffer recipes. And it'll give you a description of what is in each mini prep buffer. So generally the first buffer that you resuspend the bacteria in is just a normal buffer. It's like tris, and the only thing that's different in it is that it has RNase. So what RNase does is it degrades RNA, because these silica columns, they'll bind RNA just as well as they'll bind DNA. So what you want to do is you want to try to get rid of all the RNase. And you want to make sure that your solution is buffered. So remember, if your pH is 7 or below, what happens is it changes the charge of the DNA so it doesn't bind to the silica. So the first few buffers, what they do, the first one resuspends and has RNase to get rid of all RNA. The second one has a chemical called SDS, sodium dodecyl sulfate. It's actually a chemical that's in shampoos. SDS is what makes things foamy and bubbly, right? Because it's like a detergent. And the cool thing about detergent is that it, when you mix a detergent with oil, right? That's how you get your hands to stop being oily. Or how you dissolve oil is you mix it with soap, right? Because soap creates can bring oils into solution. Well, you imagine a cell wall of a bacteria, it's all oil, it's lipids, right? So when you mix it with this detergent SDS, what happens is the cell walls open up and they release DNA into the solution, right? So the next thing you add is you add some denaturant. So finally, what you're going to do is you're going to denature all the proteins and all the DNA. So you're going to try to make sure that all the proteins that are bound to the DNA and all the proteins in solution precipitate out. So what you get at the end of adding the first three buffers is you should have DNA in solution and protein and cell debris as that white stuff, right? And RNA should be, chopped up. So then what you do is you spin down all that junk, and that junk, that's that white stuff. You spin it down to the bottom of the cell. You take your solution, which should hold your DNA, and you dump it over the column. And then you spin it through the column. And what happens is because of the pH, your DNA should bind to the column and it'll stick to the column. It just sticks to it. Obviously, some of it will come off during the wash step. So in the wash step is basically ethanol, isopropanol, some sort of ethanol. Because what it'll help do is it'll help remove the salts, but it'll still keep the DNA stuck to the column, right? So you're washing away any sort of crap that's left over by using ethanol. You can use ethanol, isopropanol, any sort of alcohol works. So you want to make sure that you spin that extra to get rid of all that alcohol, because the alcohol will mess with future experiments. After you get rid of all the alcohol and your column is dry and there's just DNA on there, then you add water or you add a buffer, which changes the pH of that silica column, and the DNA will come off then. So that's basically how the mini prep works. Yeah, you can incubate longer in the elution step. You could add less. So sometimes I'll only add like 30 microliters instead of 50. Um, David, if you look at the SnapGene files, it should, I think GFP is like 4,500. Um, and P dusk and P dawn. Let me see. So P dusk and P dawn are a little bit bigger because they have inserts. But P dusk and P dawn are like 8,000. So yep, T. Williams is exactly what happens in the mini prep. Yeah, so right, like the more bacteria you start with, the more DNA you'll have at the end. But if you have too much bacteria, um, then what happens is you it doesn't all get lysed, right? And you end up with a lot of junk because not everything gets washed away. Yeah, afford your nuance. You can run the elution through a second time, like double brewing coffee. Um, the problem is you get a lot dirtier of a prep. So it's like if you're trying to separate the signal from the noise, the first time your signal is really high and your noise is really low, right? Because there will be a lot of DNA that will elute and a little amount of other stuff. The second time around, there's not as much DNA left over, but there's a lot of crap left over. So you're going to get, you know, more crap to DNA ratio the second elution around. So even though you'll get more DNA, generally I try to avoid the double elution. Uh, but if you're desperate, um, it's totally fine. And it really depends on what you're using the DNA for. David, the PDusk and PDAWN files aren't on Google Classroom? Is that what you're telling me? Are they not there? GFP plasmid sequence. All right, I'll add material PDusk plasmid. Oh, I know why. I know why we didn't put the sequence up there. In the first class, we downloaded. You can just download it directly using SnapGene. So if you're using SnapGene, what you can do is you can just import the files directly from SnapGene. So you should just be able to open up SnapGene, and when you open it up, it should say import, and then say add gene plasmids. And when you click on add gene plasmids, you can just search for PDusk, and you should be able to easily download it and import it into your SnapGene. So generally, Joe, the large bacterial chromosome gets broken up and sheared and turned into small pieces of DNA, and because there's so many proteins bound to it, it stays in the white stuff and it doesn't make it. But it's not always the case. So if you're doing an elution, if you see a very thick band that's kind of faint, so and it'll be like it'll look big and wide, and when you run the gel, it'll start to disappear really quickly, and it'll run really high up on the gel. As opposed to when you run the plasmids are very discrete bands, right? They're just like a line that's skinny and solid. It's not to say all the times your plasmids are like that. Sometimes if you purify a lot of plasmids, they'll run fat, but they'll also run really bright. So they'll be really fat and bright. Um, so the PDusk we're using is the same, except there's a gene of interest inserted, which is the RFP gene, or DS-RED2. So that's why it should look red or pink when you grow it up, if you grow it up in the dark. All right, what else you got? What other problems you running into? Yeah, afford your nuance. So you could add as much DNA as you want. Um, but the problem is that you use up all your sample, right? So DNA is for you want to actually use it, right? So if you run 25 microliters, then you send 10, 15 microliters off to GeneWiz to sequence, right? Then you're left with like 10 or less microliters for your experiment. So generally, you also, and it's also a good indication of how much DNA you purified, right? If you can't see the DNA by adding 5 or 10 microliters to your gel, then you haven't purified enough to use in further experiments. So even though you might want to add more just to see if something's there, it's not that useful. All right, Joe, what's your question about the next experiment? Hit me. Yeah, buzzwordy. Sometimes people will cut, uh, they'll see where the band is on the gel and then they'll cut it out and try to use that. Generally, that's, to me, I think it's like too complicated. If you have to cut out a piece of DNA from the gel, you're probably trying to do something too complicated. Okay, so the histidine tag. So generally when you put a new protein into a plasmid and you are making it in bacteria or yeast or even humans or something like that, you want to be able to purify it, right? Because a lot of proteins are used as drugs. A lot of proteins are used to be able to test. Um, you want to test the enzymology or something like that of the protein. And a lot of proteins you just want to be able to detect in some way. So what they do is on the end of a protein, they'll put this tag, which they call a hexahistidine tag, which is basically just 6 histidine amino acids in a row. Now, histidine amino acids are great because if you put 6 histidine amino acids, it doesn't exist in nature anywhere. So it's easy to detect with like an antibody. So if you're trying to detect if this is if you actually, the genetic engineering worked, it's really easy. If you're trying to detect something through like RT-PCR, it's really easy because nothing else should have a similar DNA sequence. Now, if you want to purify the protein, it also makes it really easy. Hexahistidine tags, they like to bind metals, right? So they like to bind things like nickel and cobalt and things like that. So what scientists figured out is if you have a column or a material that has nickel or cobalt on it, what you can do is you can grow up the bacteria, bust the cells open, run that solution over a column just like you would for DNA, and all the proteins that have histidine will bind to the nickel. They'll bind to the nickel. And then again, you can get them to fall off the nickel by just changing the pH of the solution or outcompeting them with a molecule that's similar to histidine called imidazole, right? So the histidine tag is just kind of like a universal tag to use on proteins that you're trying to test different things from. Can you grow antibodies in E. coli? No, usually what you do is you buy the antibodies from companies. So you can look up like hexahistidine antibodies. They're usually pretty cheap because they're really common. So you can definitely get those antibodies online. So Joe, the RFP plasmid is not present in the bacteria. So what you should be using when you do your transformations is you should be using a strain of E. coli that's just DH5 alpha, or you should have received a strain of E. coli that's just called E. coli. If you didn't receive that strain of E. coli that's just called E. coli, let us know and we'll send it to you. It might have got missed. Um, but what you're trying to do is you're trying to put this new piece of DNA you created into a strain of bacteria that shouldn't have any plasmids. So there should be nothing else in there. I mean, you probably could get away with just using one of the strains you already have as long as you plate them on opposite antibiotics. Um, it should be totally fine. But, you know, in science is always best to do the correct thing, not the easy thing, because it wastes less time. Yep, the easy thing, even though it seems like easy and possible, what ends up happening is it usually gives you a lot more headaches than what you what you hoped. Okay, Joe. Well, you know, email or message somebody, and we'll make sure you get another strain of E. coli. Yeah, all the strains are on sale at the ODIN. You should be able to get the. Well, I don't think the GFP one is. The P Dusk and P Dawn, the DH5 alpha. The GFP bacteria is available in a kit. Um, the color changing bacteria kit. But yeah, they're all available. Yeah, you can make your own bacterial stabs. You know, just make up some LB agar with antibiotic and put it in a little tube and, you know, stab your bacteria in there. So, what other methods of purification are there to use besides 6X histidine tag? There's a lot, David. You should look online for protein purification methods, protein detection methods. There are a lot. You technically don't even need a tag, but it depends on a lot. So, like, there are a lot of different methods. Um, you have some dry E. coli pellets from another experiment. Would these work fine? Um, maybe. Do you know what strain of E. coli they are? If you um, if you know what strain of E. coli there are, then I could give you a better answer as to whether they would work. Can you buy little tubes from the ODIN? Unfortunately, we don't sell the little glass tubes for stabs. We buy them on online. I'm sure you could find them on Amazon or eBay or places like that. Just look for little small glass tubes. Has anyone at the ODIN made any knockout knock-in animals? No, we have not worked with any embryos here yet. So, Joe, the strains that are used generally in molecular biology, they are deficient in a couple proteins. So, like the strains that you use for growing up and purifying plasmids, they're deficient in proteins that could damage the plasmids, certain recombinases and things. So it's not necessarily what strain you use, um, but it helps. Are we planning to do a 103 class? We're planning to do more classes. It just takes time to get them put together. So not necessarily a 103 class, but more classes working on a mammalian cell culture, mammalian genetic engineering class. So to engineer some mammalian cells. We're working on mushroom class, plant class, um, a CRISPR class. Yeah, so we got a lot of things that we're working on, but you know, it takes time. And first you'll need to complete 102, you know. You got to get this stuff working. The requirement to sign up for 103 is going to be you have to have the materials from finished from 102. Uh, the main differences are just what genes they contain. Um, so like strains that are for DNA production are minus a few genes, and strains that are for protein production are minus a few genes. So yeah, it's just scientists removed a few genes that they thought might help. All right. What other questions? Anybody got any other questions? Run that time. Yeah, you got to get these experiments done. Uh, DNA synthesis companies. Um, you're in Canada, right, David? I don't know. It's probably better to try to find a company in Canada that will ship to you, because shipping from the US to Canada, usually companies are, you know, pretty sketchy about that. So I would just look for a DNA synthesis company in Canada and see if they'll ship to you. It can be if you're trying to order something from the US, just use GeneWiz. Well, it depends on how big of a piece of DNA you're trying to order, but GeneWiz is, if you're using them for sequencing, use them for synthesis also. They're usually pretty good. So we don't allow weird purchases. I'm sorry, Joe. Just let us know, right? And we'll ship them to you if you pay the cost of shipping for the smaller stuff, because usually the smaller stuff is pretty cheap. Do we have, yeah, forward your nuance. If you look in the Google Classroom, other people have posted some good results. So check out the Google Classroom. Um, yeah, all the experiments we have validated, and also a lot of DIY biohacker scientists use the same materials, right? So most people don't have their own gel box or things like that because they're just so expensive. There's really no reason to. What you can do with your gel box is you can replace the aluminum foil with platinum wire, and you could replace the batteries with a power supply. But some people use the batteries for forever. They can last a long time. Yeah, no, Ford or nuance is totally true. Like, science is hands-on, right? And so it's like learning a guitar or something. You can't expect to be an expert guitar player after playing for one day. It's going to take a while. So what it is just doing these things over and over again, failing and succeeding and just doing them over and over again, and then getting an intuition for what works and what doesn't work, or what mistakes you made and what mistakes you haven't made, right? So it's all just into getting that intuition. Christopher, at what point would you recommend getting a PCR machine? It depends on what you want to do. You could probably get away with never having a PCR machine nowadays if you just have companies, synthesize and make your DNA for you. Um, if you want to get a PC, one of the classes we're trying to do is a class with PCR, but it's hard getting so many PCR machines. We're trying to acquire like 20 inexpensive PCR machines, but it's hard. It's hard. Um, but yeah, that's what we're trying to do. We're trying to run a PCR class and send out a bunch of PCR machines. But yeah, like T. Williams, I think with these classes, what we're trying to do is get you the supplies, basic supplies that you need. Um, and then as you go along, every now and then you could upgrade, right? Upgrade to a better gel box, upgrade to a better power supply. Afford your nuance, OpenPCR probably works pretty okay, but it's also $600. I think that's unreasonable to ask for somebody for a PCR machine. Um, It's not worth $600. It's a piece of wood with a heating unit in it. So if we had a PCR machine for $600 that we were selling to people, we would have to make money off of it. So we'd have to sell it for 800, right? And then all the rest of the materials for the class would probably run it over 1,000. And I think that limits the class. Who's going to be open to the class, right? So what we're trying to do is make the cost inexpensive by getting refurbished and used PCR machines for like $150. So then we can have a class that probably runs, I don't know, 750 bucks, and you get a PCR machine out of it. That would be pretty awesome. But you know, we're collecting PCR machines. It's taken us a bit of time. What would I say are the one or two things you need to accelerate the growth for ODIN? Right now, we just need more teachers. We need people who can teach these classes. We need people who can write curriculum. It's really hard to find scientists who who are willing to do that, even for pay. Um, and it's hard to find people who understand how to do stuff in a house, right? So a lot of scientists I talk to are like, "Oh, I can teach a class," and they're like, you know, they want to teach a class with like $1,000 million pieces of equipment, and it's like, no, you can't do that. Josh, I think we talked about it. If you could build gel boxes, let us know. Let us know your price. We're probably willing to pay about $75 a gel box. We can even supply all the platinum wire, right? But if somebody can make a gel box that's, decent condition, has plugs, so people can plug in banana plugs or leads, install the platinum wire. Yeah, just let us know once you finish your master's degree. We're open to people helping us out. Yeah, Joe, I mean, it's hard, right? It's just hard in general, getting people who understand. Well, if you make a gel box, let me know. Let me know if you make a gel box. Make one and send me pictures. I'll send you some platinum wire. You install platinum wire, and then I'll pay you for it. We'll test it out. If it works, then we'll order some from you. Make a prototype. And understand that, like, if we order 20 from you or something like that, we're probably going to want it within a week or two. So, all right. Any other final questions before I take off? Again, keep doing the experiments. And if you have trouble, make sure you post your pictures and stuff in the, Google Classroom, because that's the only way we can help you out. And don't feel like, there's anything wrong with not being successful, not having success. Like, it's really hard to do science. Don't expect to do it correctly the first time. It's okay to fail. We're here to help you. We're here to help you do it correctly. So, post your pictures. Make sure you keep track really, really good of the experiments you do, so then we can help you troubleshoot. And let's get this done. we already have some people who had some successful sequencing results. Now we just need some people to get some successful genetic engineering. All right. So I'm counting on you to make this class a success. Okay. All right. Have a good night. I hope you feel like Thor. It'll be great. All right. Have a good night, everybody. I'm out.